next we will be looking at allosteric inhibition. Right, so allosteric inhibition is specific for a type of enzymes known as allosteric enzymes. Right, so before we look at inhibition, let's look at what are allosteric enzymes. Okay, so allosteric enzymes are usually consisting of more than one polypeptide chains. Okay, and each polypeptide chain form a subunit of the enzyme, and each subunit has its own active site. Okay, so allosteric enzymes are usually quaternary structures with more than one subunit made up of more than one polypeptide chains with more than one active site. Besides the active sites, right, allosteric enzymes usually also have allosteric sites, and these sites are usually located at where the polypeptides are joined together as they form the one allosteric enzyme. Okay, so and usually, right, the allosteric enzymes alternate between two conformations. We have the active conformation or active form, which has a higher affinity to the substrate, and an inactive form, which has a lower affinity to the substrate. And this alternation between the two forms can be regulated by the binding of substrate as well as binding of allosteric activators and allosteric inhibitors. Okay, so let's look at how the binding of substrate, right, regulates the change in the conformation of the allosteric enzyme. Right, so substrate binding to the active site of one subunit of the one enzyme molecule would change the conformation of the entire enzyme molecule to the active form, and that makes the active sites on the other subunits of the same enzyme molecule to be of a higher affinity to the substrate, and that facilitates binding of the substrate to the other active sites. And this phenomenon is known as cooperativity. Right, so this is represented in this picture that you have in the notes. Okay, so if the enzyme was at first in the inactive form, right, it has a lower affinity to the substrate. Lower, right, not zero. So the substrate would still be able to bind, just harder to bind, right, or less likely. But once a substrate has bound to one of the active sites, then it changes the conformation of the entire enzyme to the active form, and that allows the other active sites to receive substrates more easily, right, because it has now a higher affinity, right, for the substrate. So this is again the cooperativity. Right, and because of this phenomenon, right, the graph of rate of reaction in increasing substrate concentration will show a sigmoidal or a S-shaped curve, which is a hallmark of allosteric enzymes. Okay, so for the normal enzyme, the non-allosteric enzymes, from what you have learned in lecture 2, right, this is the very nice and smooth curve that shows the increase in the rate of reaction when the substrate concentration is increased. But for the allosteric enzyme, the increase at first in low substrate concentration is slower, because most of the enzymes are in inactive state. The affinity for the substrate is very low, right? So in the same substrate concentration, the rate of reaction would be slower, right? It is less likely to form the enzyme substrate complex. But the increase in the rate of reaction would be very fast in this portion here. That's because of the cooperativity, right? So when there are enough enzyme molecules that have been bound by at least one substrate, then the enzyme molecules start to, more of them at least, start to change to the active form, right? And that has a high affinity to the substrate, right? So the increase in the formation of the enzyme substrate complex would be much faster when substrate concentration continues to increase. And this increase will slow down, and eventually there would be no increase when the enzyme becomes saturated. Right? Even though they are mostly in the active form, active state, but they start to become saturated in very high substrate concentration. So the increase will slow down again and eventually become constant. Okay, so this gives a sigmoidal curve, and again that's a hallmark of a allosteric enzyme. Alright, so having a look at what is an allosteric enzyme, right, let's now look at the allosteric activators and inhibitors. Right, so they too, right, would regulate the change in the conformation of the allosteric enzyme. The allosteric activators, right, bind to the allosteric sites, and that would cause the change of the enzyme to the active form, while the allosteric inhibitors, once it binds to the allosteric sites, it causes the enzymes to change to the inactive form. Okay, so if you look at the allosteric inhibitors, right, it binds to not the active site, right? It binds to a site other than the active site, which is specifically known as the allosteric site in this case, and it causes a change in the conformation of the enzyme such that the active site is, well, less able to bind to the substrate, right? So if you look at this mechanism, right, it is similar to the non-competitive inhibitors, right? So allosteric inhibition, right, therefore can be considered as a subset, a form of non-competitive inhibition. But that's why we look at the learning outcome, right, under the non-competitive inhibition, it says bracket, including allosteric inhibition, right? So that's why it is phrased like that. Okay, the mechanism of allosteric inhibition, right, with the binding of the allosteric inhibitor to the allosteric sites, changing the conformation of the enzyme, changing the shape of the active site, resembles or really is very similar with the non-competitive inhibition that we have looked at earlier. Okay, so an example of allosteric enzyme and allosteric inhibition would be the enzyme Phosphofructokinase, which is involved in cellular respiration, and more specifically in the process of glycolysis. Okay, so it is inhibited by ATP, which serves as an allosteric inhibitor when ATP concentration is high in the cell. Right, so it would inhibit the activity of this enzyme, therefore slowing down the rate of glycolysis, which in turn slows down the rate of respiration, because there's already enough ATP in the cell, right? High ATP concentration. Right, so it prevents the further respiration to produce further ATP when it is really not needed by the cell. Right, so this is a very good example of the allosteric inhibition, and anyway, you will learn about this enzyme again next year, right, when you learn about respiration. Okay, so this is like a heads up for you, yeah? Okay, next we will move on to the end product inhibition. Okay, so before we look at the end product inhibition, we need to look at what is a metabolic pathway. Right? So a metabolic pathway is a series of enzymatic reactions, each catalyzed by a different enzyme. Right? So as you can see in the example given in the notes, right, which I have it here, this is a metabolic pathway that includes 3 enzymes. So the first enzyme catalyzes the formation of intermediate A from the initial substrate. The product of the first enzyme becomes the substrate of the second enzyme, and so on, until eventually the end product is formed. Okay, so a common phenomenon that we see in such metabolic pathway is the phenomenon of feedback inhibition, in which the end product, once it is of high enough concentration, acts as an inhibitor to one of the enzymes in the pathway. Right, so as you can see in this example that you have in the notes again, right, the end product in this case serves as an inhibitor of enzyme 1. Right, so therefore the initial substrate is not converted to intermediate A, and the entire metabolic pathway stops its function. Right, so that would prevent further synthesis of end product when the end product is already of a high enough concentration. Right, so this helps to prevent wastage of resources. Right, you don't need more of the end product, so you don't need to waste the substrate and convert it to the end product, which is not needed. And maybe some of the reactions might require energy, right, in the form of ATP. Right, so that would, by stopping the metabolic pathway, that would prevent this wastage of energy as well. And also, right, when we have enough of a particular product, then some of the intermediates in the pathway may be able to be channeled to other pathways to form other useful products. example is this pathway here that you have in the notes as well. Right, so when there are enough product D, it may be able to inhibit enzyme 2. So intermediate B, which continues to be produced by enzyme 1, can be channeled to another pathway to form product F. Right, so having enough of product D, which stops its own synthesis, but that still allows for product F to be produced. Okay, and of course, again, by preventing further synthesis and accumulation of the end product, it prevents any potential adverse effects that may be caused by having too much of the end product. Okay, so looking at the question that you have in the notes, right? When you look at this example in figure 6.10, the end product acts as an inhibitor to enzyme 1. What kind of inhibitor is it? Of course, it's an end product inhibition, right? But what kind of inhibitor is the end product acting as when you focus on enzyme 1? Right? It binds not to the active site, right? It binds to a different site. It causes the active site to change in shape, as you can see in the picture. What does this remind you of, right? So it is actually an example again of non-competitive inhibition, right? The end product itself, right? If you look at in context to enzyme 1, right? The end product serves as a non-competitive inhibitor to enzyme 1, and that stops the function of enzyme 1, as you can see in the diagram. Okay, so even though there's no mention of the end product inhibition in the learning outcome, right? I have decided to still show you an example because the mechanism of how the end product inhibits the enzyme is similar, or is in fact the same as a non-competitive inhibition. And so it can be an application, it can be an example of how non-competitive inhibitors work in our cells in the context of a metabolic pathway. Right? So with that would be the inhibitors